Hi. So in this video, I'm going to be talking about nucleotide polymerization. So nucleotides polymerize just like amino acids, which is in a condensation reaction. So water is going to come out. And so remember with proteins, we had an amino acid linking together with another amino acid in a peptide bond, and water came out. And so in nucleic acid polymerization, you can just replace those with two nucleotides, and they're going to come together and water is going to come out. And in proteins, right, we had a peptide bond, and in DNA, the bond that forms is called a phosphodiester bond. So what does that reaction look like? So what's going to happen is that a phosphate group on the 5' carbon is going to link up with a hydroxyl group on the 3' carbon. So these two are going to come together, and I said it was a condensation reaction, so water is going to come out. And so here I'm showing a deoxyribonucleotide, but this could also occur with RNA, in which case there would be another hydroxyl group here. But that doesn't affect the bond that forms, this phosphodiester bond that forms between these two groups. And so here we're going to number our carbons, so 1, 2, 3, 4, 5, and we're going to have this 5' phosphate group come together with this 3' hydroxyl. And water is going to come out because it's a condensation reaction, so there goes water, and there links up our phosphate, and there's our this is right here, that line is our phosphodiester bond, and this forms the primary structure of both DNA and RNA. And what it creates is this sugar phosphate backbone. So sugar phosphate, sugar phosphate. So you have that backbone, and then inside the molecules, right, facing a different direction is the nitrogenous bases. And what it also creates is a molecule with directionality. So like amino acids linking together gave us a peptide with directionality, we now have a nucleic acid with directionality with the 5' carbon at the top, and then the strand's going to grow at that 3' carbon on the bottom. So the next nucleotide is going to come in and join right here. So we have this 5' to 3' direction. And I just want to point out now, this might be what's opposite of intuitive, because you always want to say 3, 4, 5, but it's the opposite. So it's 5' to 3'. So here's just an example of a growing molecule. So before I just linked together two of these, right? So here's one phosphodiester bond, and then here's another one, and another one. So here we have a growing strand, and then the next nucleotide is going to come in and join up with this 3' hydroxyl group. And so this can be represented also in a line drawing. So here's our 5'. So this line here represents our sugar phosphate backbone, and the molecule is growing in this direction. And then these ticks off that backbone represent the nitrogenous bases, and they can be represented with just a single symbol abbreviation. So this growing strand, right, we're going to have another nucleotide is going to join at the bottom of that molecule at the 3' hydroxyl group. But that polymerization is catalyzed in real life by enzymes. And the energy for polymerization comes from adding 2 extra phosphate groups to those nucleotides. And so here's an example of what an activated nucleotide looks like with those extra phosphates. And so those phosphates have these negative charges on the oxygen, and that's a lot of negative charges to be compacted into one area. So that stores a lot of potential energy, and because of that, the cell wants to dissipate it. And when it dissipates, you can release energy. And we use that energy then to link on more monomers to our growing polymer. So to summarize, polymerization occurs via a condensation reaction at the 3' hydroxyl to the 5' phosphate. The polymer has directionality in the 5' to 3' direction. And the process of polymerization requires energy because it reduces entropy, right? When you're linking together monomers, you're decreasing the amount of entropy or disorder in a system. And it's catalyzed by protein enzymes, and it requires activated nucleotides. And so let's kind of put all this together in a real life scenario. So there's a technique we often use in the lab, and it's called PCR, or polymerase chain reaction. And it was developed in 1983 by Carey Mullis. and he shared the Nobel Prize in Chemistry in 1993 for his discovery of this technique. And what this technique allows us to do is it can help us copy or amplify a gene of interest. So we're actually able to study an individual gene, and we can ask questions about mutations or if it's there or not. All sorts of questions can be answered using the technique PCR. But that's not what I'm going to point out about it. What I'm going to point out about it is all that you need to add to a tube to have a PCR reaction. And what you need to add is your gene of interest. So you just get DNA out of a cell, and you need to add Taq polymerase. That's the enzyme that I kept mentioning was important to create the polymerization reaction, or to allow the polymerization reaction to occur. And then you put in deoxyribonucleotides that are the activated nucleotides. Okay, so they're just listed here, right? So this is the activated adenine, the activated cytosine, guanine, and thymine. And then you just put in some buffer. So all you need to do is just add your nucleotides, your enzyme, your gene of interest, and bam, you can amplify a gene. And so we can do this in a test tube. So that's just kind of tying together everything about polymerization. And then the next video, I'm actually going to talk about the double helix of DNA and a little bit about the other structures that RNA can form.